the semester. We're very lucky to have a special guest with us today, Dr. Juergen Polle from Brooklyn College and City University of New York. He's in the Department of Biology there and has been working with algae for a very, very long time. Once we get to the stage, we sometimes don't prefer to say exactly how long, but it's measured in decades. So, anyways, he came in yesterday. We had a very good chance to talk about all things biological related to algae. And his talk today is going to be a little bit more of an applied focus, looking at microalgal biofuels, which many in the group here have an interest in. And if you have an interest in biofuels in general, I'm sure that you'll be able to learn some interesting things. Dr. Polle, before he was at the City University of New York, was at Berkeley, working in the plant in microbiological biology department. And he was there for about 5 years. And then before that, he was in Germany at Göttingen. How do you say it again? Göttingen. Okay. Fortunately, he hasn't quizzed me on my German skills. That goes back a long time. But in any case, he was there where he worked on his PhD in the biology department and has been working with algae originally more from a fundamental perspective and the photochemistry and the photosynthesis process, but then more recently has been trying to apply that to the critical national and international needs for biofuels. He's a member of the National Alliance for Advanced Biofuels and Bioproducts, which is one of the major DOE projects that is trying to advance algae biofuels. He's in the algal biology portion of that particular project, and I'm sure that he'll touch on some of the findings that were associated with that. He's also been a foundational researcher on some Army, actually Air Force projects that were also looking at algal biofuels. And so with that, I'm going to go ahead and turn it over to Juergen, and you got your second not to use that, or your
I can try without, I think.
Okay, and then as usual, we'll pass around the sign-up sheet, so if you wouldn't mind trying to juggle that with your pizza and everything else, and go ahead and sign in for today's summit. Okay.
Thank you very much for the invitation. If you can't hear me in the background, speak up. I think we can take questions in between, short questions, but maybe should save major, longer time requiring answers for at the end. And you're looking at from good. Okay, so I'll just give you an overview, background, energy, algae, how that fits together, a little historical background, a little background on the algae. What I want to talk about mainly is the strain discovery work that was funded by the Air Force for almost 4 years and is continuing through the Department of Energy funding. And from that I go a little bit and touch, I have 2 or 3 slides at the end, metabolism. So, but because I decided to have more introduction, I have to cut at the end. But we'll see. And then, brief summary. So if we go further, the major sources of energy that we have, I've listed here. What we're looking at right now is to get bioenergy, get energy basically to replace some of the fossil energy sources that we have. And what we're trying here, we try to grow biomass. That's what it is all about, using algae ultimately. So then go to the next. What do we have as what's coming out? These are basically the ancient hydrocarbons that we're looking at, gas, coal, crude oil, petroleum. I have a little bit of the chemistry here. What are the carbon chain lengths, for example, in the oil that we get? And what we are basically looking at more specifically is replace the transportation fuel. You can't fly jet fighters on hydrogen right now, and you can't fly them on electricity. So we still need liquid transportation fuel as oil. If you look at some of the general projections here, this is from the 50s, what about oil? Where's the oil coming from? Well, there's, at least for the US, a lot, well, not in the beginning, but if you look here, the 80s, lots of it. The blue curve is the net import of oil into the US. The yellowish curve is what's being produced in the US, or what has been produced. So from the 50s up to about 3 years ago. And then here we have the red curve is the consumption. So anything that's not produced ends up as an import. You can see here it's going first oil shock and so on. So there was recovery, but steady again, used increase of imports. If you calculate, so the next one, if you think about how much the US spends in terms of dollars every single day, what we pay combined for oil imports, that's just money thrown out of the window, if you ask me, but it's what, about 400, that's an estimate, depends on where the dollar per barrel actually is. So this is low if we talk about today. This was calculated with a low dollar value. Billions of dollars per day. And I want to bring that out here because we have to look at how much money is actually given for research. And research for energy. We spend a lot of money just to pay other people to give us oil. And that's per day. And I don't think anybody has spent a whole lot of the fraction of that for research that can help us get further. Okay, just to put things in perspective. Money for research is here, and money for oil going out is that per day. So, if we move on, so if we look at the oil imports and the barrel value, we're about somewhere here. And, well, in earlier times we were much less. Move on to the next one. So there's a lot of money that's going into other people's pockets. Now the biomass that I mentioned is basically coming through renewable sources and, for example, through plants. Plant biomass. And plants for energy is not a new idea. If you go back, there was an Italian scientist, here's 1912, I hope you can read that. He published in Science, in the journal Science. He came to New York, gave a talk, and then a few years later it was published. But in the early 1900s here, he gave a talk and he said, well, the arid lands, there will spring up industrial colonies without smoke and without smokestacks. Forests of glass tubes will extend over the plains and glass buildings will rise everywhere. So where are the glass tubes? This was 100 years ago. 100 years. And what is he talking about? Photochemical processes. Meaning plants. Photosynthesis. Actually, he was working with algae. In addition, not just plants. But I mean, this is 100 years ago, and he's talking about weaning us off the fossil fuel, basically. And using biological processes. But what was the fuel? Then it was coal. And soon after they published that, they found something cheaper, and it was the oil that went everywhere. So now we're talking about getting us off oil and going back. So it's not new, especially for the students. It's all coming back in cycles. You just have to live long enough. So then the next one. So the ideas are not really new. And even algae is not a new thing because it's coming in cycles. MIT, 1950. MIT here, the people are building algae reactors on top of the roof. All that was repeated in the early 2000s. So not 100 years, but 50. So it's cycling. There are not too many new ideas in that field around, I think. There are new ones. So what is, if we talk about biological sources for energy, so we talk about bioenergy, which means renewable energy from materials that are derived from biological sources. And what do I mean with that is, the example is corn to ethanol. So here's my biological source, and the material is then the ethanol as where the energy is stored. We have corn to ethanol, we have, when you go further, sugarcane to ethanol, we work a little better in certain places. But now what I want to talk about is algae to oil. And what I talk about are microalgae, microscopic algae. So, talking about algae, then I hope everybody knows what algae are, but if not, then let's go to the next one. We have on one side microscopic algae. You need a microscope to see them. Of course, the water is green, there's something in there, but you need a microscope to see them. Versus macroalgae. This is from the coast of California, where we have kelps that are dozens of feet high, but underwater forests, basically. And they're several meters long, so here's just the scale, 1 meter over 10 micrometers. And what you have to keep in mind, and maybe that comes out here already, there's great diversity within. Someone talks about algae, that's not just one small group of organisms. We talk about plants, it's a small group in comparison to the algae. We have very diverse organisms. So when you go back, further, the diversity of the algae comes through what we call then, if you believe in evolution, primary endosymbiosis, tertiary endosymbiosis, secondary endosymbiosis, tertiary endosymbiosis, plastic replacement event, whole shuffling of organisms. One ate the other, ate the other, ate the other, and things happen in between. So when you look at plants, in the strict sense, you're basically looking here at this part. The green algae are here. Then you have the red algae and what we call the glaucophytes. 3 major branches of algae, 1, 2, 3, coming out of primary endosymbiosis. A eukaryotic organism taking over a cyanobacterium. Now, over time, so here's the cyanobacterium, primary endosymbiosis here. Over time, if you go further, there were other organisms that ate the red alga. And the photosynthetic process was maintained. And we have, for example, here, heterocontrophytes are diatoms, the big kelps, brown algae. Here are haptophytes, cryptophytes, dinophytes. There are some organisms that belong into the group of algae, but they don't perform photosynthesis anymore, and that's they found a better host. The malaria parasite, for example, still has a remnant of the chloroplast, but it's a parasite. So there's a whole group of that as well. And out of the, go a little further, there is secondary and tertiary endosymbiosis. Here, secondary. In the green, for example, Euglena, and then another whole group, chlorarachnophytes. So in terms of diversity, these are the plants. And we're as far away in terms of relation from the plants as some of these organisms are from the plants. That we have to keep in mind. So there's great diversity. And that goes back to how they grow, what will they tolerate, how much light, and so on. So various groups of successful or less successful algae. My focus in the past was the green, which are closer related to the higher plants, but there are significant differences in the metabolism of the green algae over the higher plants. And the next, where do algae live? Well, I traveled a lot in the past years, going everywhere sampling. So this, for example, the Brooklyn Botanical Garden, Microcystis, Cyanobacterium, a nice layer, almost monoculture here on the freshwater pond. This is a photo of the salty sea. You can see a little brownish color. Here's some green algae, but the brownish color, lots of diatoms. Oh, and this is my favorite here, hypersaline. This is salt crust. This is saturated salt solution, the Bay Area, off of the Barton Bridge in California. What you see here is orange, mainly Gonaliella salina with some other Gonaliella halophyte algae that can tolerate saturated salt. There's some algae that can grow at very low pH, there are algae that can grow at very high pH, anywhere in between. At temperature profiles, there are algae in water bodies, there are algae in lichens everywhere, there are algae all over. You start off with algae products in the morning, even though you may not know it, and you end up with algae products before you go to bed. all throughout the day you have products that you consume right now that have algae material in them. You just don't know it because nobody ever cared. But this is now for energy, not for other products. Okay. What are the production systems for energy, bioenergy? How do we produce the algae biomass? Well, in the past, what? 5, 6 years, all these little companies came up, some made it, some didn't. The list got longer and longer, and here are just some of the maybe major ones in the US. If the company's not on the list, okay, I took a few. I categorized by different kinds of technologies. What you see down here on the lower right is what I call the dark side versus the light side. The dark side just means they use fermentation technology. They use algae as the organism, but they feed them sugars. No light. So fermentation technology. Zobaza Martek is the older company that already existed in the past, and now Solazar. We have maybe a few more, but these are representative examples here, especially in the US. We have photobioreactor technology. And I've seen photobioreactors indoors and outdoors and different systems and tube systems and trough systems and very different ones. So here are a few companies that claim they work or will work with photobioreactors. But I think the vast majority of the companies is somewhere in an outdoor open pond design system. Or, what I didn't put in, there are some companies that claim they will go out there and harvest the natural blooms. That is not on the list. But a lot of companies claim they will go the open door approach. The most prominent that you may have heard about in the past are Sapphire Energy, located in San Diego, Synthetic Genomics, together with ExxonMobil, located in San Diego, just up the street of Sapphire. Symbiotic is the one that's shown here, courtesy of Dr. Van Amotz, who is at Symbiotic. They use the chimney of a coal-fired power plant to feed the algae ponds. And they just built a fairly big facility in China, I think in the Qingdao area. So, those are different production systems. And all these systems have different kinds of algae. If we go further, the objective now, where does my research come in? Strain development means we're basically looking at pond to plane. That's from the Air Force here. This is the first plane, I think, run one complete engine on a fuel derived from algae oil. So one engine complete, that was in last year, 1.5 years ago. So, but all these productions, what people do and need are new strains that go into their whatever production system they have. And almost all the companies first have to look at, well, which alga do I work with? They're so diverse. Do I work with green, with diatoms, with cyanobacteria? So new strains are needed for the growth, especially, I said nothing so new, but there 50 years ago they had systems where they grew algae. The problem is, growing algae was not cost competitive with an oil price of what? $20 per barrel. It gets cost competitive, we talk about $300 per barrel. Well, that was in the past when we started all the algae biofuels about 5 years ago. It's about $300 versus 20, but now we were in between over 100. So then the ratio gets much better. But still, the productivity of the system needs to be improved. And I think many here are engineers. I don't want to downplay the engineering side. But when I look at this from a biological perspective, the largest gains, I think, it's my opinion, are in the biology. Yes, you can make gains through engineering, but the largest gains possible are in the biology. And I worked on the light harvesting system first, and So I think that's still a valuable option. But we need new strains. And new strains means either through, we can go further, we find new ones, we go out there everywhere, isolate from natural habitats, or we take existing algae that somebody had somewhere, and we improve the strains for, like corn. We look for specific traits, larger kernel, whatever is necessary. In part, that can go through breeding, traditional breeding. Now, many people say you can't breed algae except for Plumetomonas. That's wrong. It's just that there wasn't enough money invested to follow up on many of the genetic systems that have been reported in the past 100 or more years. So a lot is in the literature. The other part is the more newer version, the genetic engineering side. And there are multiple things we can do. We could, for example, look at metabolic engineering, make new metabolites, make more of a certain kind. We could work on light. It's all, if we talk about photosynthesis, it's all about light and light conversion. And I've worked for many years then on the light harvesting side. Or we could go to crop protection. We talk about algae large scale, and now I'm not talking about fermentation technology, I'm talking about going outdoors. It's like agriculture. You think about acres and acres and acres, it's like agriculture. And you have to think about protecting your algae from anything that may come in and be adverse. Okay, the next one. So my work focused on was the part number one, going out there, looking at, can we find new strains that are good basis to then move on with them. And the whole strategy is random. I didn't go into targeted. There are different strategies to find new algae. I can say, okay, there are specific habitats that I look at, certain extreme habitats. Or I look for specific algae to begin with. But what my approach was, go out there, isolate from as many different habitats as possible, as many different strains as I can get, and then screen those strains vigorously for oil production, for oil productivity, and specifically for triacylglycerols. So the first part of that is going out there, traveling, sampling, or finding people that send me samples. The second part is the isolation, then very rigorous screening, and once you find candidates that are promising, that grow well, maybe have lots of oil, we need further analysis in the lab. Well, then the whole thing comes down to ultimately testing strains for growth outdoor. And we had a large algae project in the 80s and 90s, the aquatic species program, and they did something very similar. Finding new strains, they came up with about, what, 3,000 strains, of which 300 looked promising. But when they took some of these and put them outdoors, they ultimately didn't perform too well. And so the major criticism was, yeah, you're just working, and come up with strains that don't make it outdoors. But I think we have enough data that says, yes, there can be strains that may be promising. But it's a pipeline. You have to go through thousands in order to find one or two or three. And testing ultimately, and this is now, I started this up to here through an effort-funded project, and then the NAP came along about 2 years ago, 1.5 years ago, we got the funding from the Department of Energy. We do this now, and a major focus is on the analysis of strains, and we have test bed facilities. Dr. Riesemann shown here has little paddle wheel ponds in a greenhouse for testing. We have outdoor facilities in Pecos, Texas, and also in Corpus Christi in Arizona, so multiple different sites. Okay, but that's a really important part at the end, that whatever comes through the pipeline needs to go outdoors for testing. Will they ultimately grow in a pond? That's what it comes down to. Can have a great lab strain. If I can't grow it outdoors, it's worthless. Okay. Where do I go sampling? So let me walk you through. The major efforts were in the beginning here, Utah. We had in California, sites, New Mexico, Texas. Several Boy Scouts that sampled here in the greater Houston area. And then, of course, where I live in Brooklyn and in around, there's lots of water. So those were the major focus areas. And why, if you look here, it's mainly the Southwest. You see the big void up here. Because when you put out the proposal, people say, where will the algae be grown? Say, they will be grown somewhere down here. That's one opinion, but an important opinion, it seems. There are people that say, well, we can grow algae here, no problem. So we're a little higher up, but the focus was for that reason down here in this belt, because people said that's where the algae will be grown outdoors, in ponds. Okay, go further. What are the different habitats? Well, it goes from shallow bird box, that can be temporal sometimes, very extreme to, oh, yeah, that was just one example. Travel companion once in a while, you need that, you never know what happens. Here are the tubes that have been going, and sun lotion, because those areas are extremely sunny. Okay. Sometimes can be difficult. I never saw any, because I'm probably trampling around. I never saw a rattlesnake, nothing, never. So, but some of the areas were not simple. So, going out there from various places, get samples, bring them back to the lab. Now the question is, how do you then look for the strains? As I said, I didn't discriminate. Some people discriminate for looking directly for high oil containing strains out of the original sample. I said I take the opposite approach. I say whatever I can get, I will test later. So first is isolation, and I mixed traditional isolation with high throughput cell sorting. Why the traditional, the combined approach? Because I noticed that when you go in and you do cell sorting on these original samples, you lose a lot. And I mean a lot. It's only the robust ones. Cell sorting means that when you see the cell sorter, it's high pressure, that little cell goes through and is damaged at the end, if it's not a robust chlorella type. So a lot of the cells do not like cell sorting, and then you lose. So what I do is traditional first plating of different media, solidified here, agarose, then we pick colonies, those go into liquid, and then, next one please, and then from the liquid we go into the cell sorter. And sometimes we're good and we're already unialgal. Only one algae here, you can see the population, 1, 2 algae populations, and P3 are all the bacteria that were coming with the algae. We played, again, back on agarose solidified media for various reasons. One is you can easily spot whether it's a clean alga, unialgal, whether it's even exenic, whether they're bacteria or not. You can see easily here. You can easily tell differences in colony size, colony color, colony appearance. That indicates that you have different species in the mix. And then we pick one of each, as many diverse ones as possible. Each one of those is a strain, but many of those will be ultimately the same. So we try to avoid picking too many from the same appearance. We want as much diversity as we can get. Okay, then the next one, we take that and put into a screening process. And the first level screen is just batch culture, measuring growth by optical density and determining the lipid content, also with a fluorescent marker, which is Nile red. That was the beginning. I got last year at this time a UHPLC system with a mass spectrometer, so we can do now metabolite profiling. That also works very well, and the tar can replace the Nile red screen. But we still grow them in batch culture, and we follow them. We pick the best out of that screen, and then we go and try to improve the media somewhat. We have 3, 4 different media. That's the second level screen, and sometimes the algae came up on one medium, but that doesn't mean it's the best medium for growth. So there's a multimedia screen in between, And then from that, we go into strain analysis, which means at this point we have bubbling columns and a water bath to keep the temperature constant, and then we can change conditions. And from that strain analysis, we basically get ash-free dry weight, so the real biomass, and we do a lipid extraction. And then we can get growth curves based on biomass and actual lipid values. And the best ones here then were moved on already. We sent 3 strains out to Texas. Freshwater strains, we sent out, I think already about 6 strains, marine, saltwater type, to a different facility, both in Texas. Okay, and how does it look? The first screen is batch culture, 100 some at a time, grow about 4 weeks, growth period here, and twice or once a week we go in, measure optical density, and determine with NIRAD the lipid content. You go next. So we change the cultures, we use NIRAD and a plate reader to get us OD values, optical density, and NIRAD fluorescence values. We correct that, normalize, and I compare to 3 different algae, and the most important one that we compare to is Chlamydomonas reinhardti. Some of you may have heard that. Unicellular green, model algae. It was not known to make lots of oil. So we want something that's better than Chlamydomonas. The next one here is now Chlamydomonas on average with NIRAD signal 4 weeks. 1, 2, 3, 4, 1, 2, 3, 4. So over time, when the culture goes from the beginning into stationary phase, we take the reads. On average, Chlamydomonas doesn't show much. So what we're looking for is a strain that when it grows and goes ultimately towards the end of the growth phase, accumulates oil. And here we have one. That was the best candidate that came out of the first Air Force project. Out here to the right is the average of, I think, about 50 strains. It's just averaged. Every single strain was looked at. What's the average that you get? That's the average. So we will look for something that has a much higher NIRAD fluorescent signal, which is normalized for growth. This one was thrown in because it's relatively low, and it would not pass the NIRAD screen. I wouldn't pick this as a candidate to move on with. But then I noticed something strange, is I find those that don't show NIRAD signals, but when you look at the culture, they turn orange-reddish. Since I'm a biologist and I know a little bit about this phenomenon of turning color from it, there's a green algae that turn in color, that means they accumulate carotenoids. And what do we know? That the carotenoids go with lipids, with trisucrosyl. So this strain must have oil, but it's not showing as NIRAD. So if we go to the next slide, this is, next one. This is the, sorry, go back. So this is the strain here, the NIRAD signal by microscopy. You see it's loaded with little, each dot here is an oil body that's labeled with NIRAD that's fluorescing. So these cells are full of oil bodies. That's this one here. Now if you look at that, there are lots of strains that turn orange. And it's one of these here that's really orange, nice. And you can only see by microscopy that they're full of oil bodies. But the NIRAD is not going into the cell. They make cysts, walls, thick walls, and the dye is not going in. That's why they don't show up in the screen. But you can do a visual screen. Whatever turns in color like this accumulates oil. So the ones that, but almost none of these show up in the NIRAD. So we have NIRAD problems. And even Bodipi doesn't necessarily go in. You need to do special things. Okay. The other part was we extract a little sample, a milliliter, spin down, extract the oil, and then run it through UHPLC, so chromatography followed by mass spectrometer. This is just the triacylglycerol standards to tell you can see the peaks nicely of the different kinds of triacylglycerol molecules. When you go now and look at the trace here, you see all those peaks behind you. This is from oil, from triacylglycerols, whereas these peaks are from membrane lipids. So we're looking for something that has lots of triacylglycerol over the membrane lipids. And so this is a very good strain here. And with the mass spec, then we can go in and we can say, what kind of triacylglycerol is this? What kind of saturation grade does it have? How long is the chain length? Okay. And these are all the triacylglycerols here, and then these are the phospholipids. This is now a photo for the example, the secondary multimedia screen, and you can see some algae grow really well in one medium, but they don't really like the others. Some are more universal. Regardless of what medium you throw at them, they grow reasonably well. So we have all the different kinds of in-between. Here, this medium was the best one. Here it was the worst. So it depends on what kind of nutrients you give the algae, how well they grow. So we manipulate already a little bit. And from that we know then which medium to take for what you see here, examples for those bubbling columns that we start. And just by visual inspection, you can tell that there are some strains that don't really grow too well. They look good in your screen, but when they go into the bubbling column here, they don't do too well. And so we have dozens now put through the bubbling columns, looked at biomass growth. This is ash street dry rates in grams per liter. And we actually get some strains that give us, here's one that kept going and going and going, and even after a few days, after a week, it was over 6 grams per liter. So that's fairly good as density. So, but this lets us look at how dense can a culture get, how fast can it do that, very fast or very slow. Some didn't grow at all. And then we pick the best ones here. And at certain times during the culture, we extract to say how much, what's the total lipid content. Then we go to the next one here. So that was the day that we had extracted for oil. Okay.
Next question.
Yes.
Is there a specific biomass target you're looking for in terms of grams per liter per day or per?
Oh, so the question is there a specific target? And the answer is yes and no. For the NAB, the alliance that I work with now and trying to find new strains, we have a target ultimately after 3 years. We want strains outdoors with 20 grams per square meter per day with 50% lipid. That's a target. Now it's difficult to say, will these bring that? And the correlation lab to outdoors is, we need more strains to tell us. But if they're not performing indoors, they won't perform outdoors. If they perform indoors, you have a chance that they will do outdoors. So ultimately then I can look at the productivity, and it's not about having a strain that has 50% lipid, but it doesn't grow. You have something that grows and produces, and there's an inverse relationship, and that's how I got my funding, because usually oil production in terms of triacylglycerol production means that's happening under stress. Whenever the algae are not happy, then they do that. And I looked at stress metabolism with Dunadilla salina, and it's very similar here. So this just plotted the daily, from the growth curve, a different way to show us how much biomass did that culture accumulate on a particular day. So here are the different strains, and the different colors is the first day, the second day, and then it crashed. Good production, crashed. This didn't grow at all. And then here we have some that produced fairly good amounts in terms of biomass productivity. That's the productivity. And we pick then the best ones, and we say, what is the lipid content? So that we can then plot a lipid productivity. And actually the ones that didn't grow here, we didn't even extract any oil. So then we come up with something like this one here, strain number 8 was more than a gram of lipid within the 5 days. So going back to the question we just had, we need a gold standard strain that we compare to. And within the consortium was decided that we have 2 production strains that already existed, for example, for aquaculture. Nanochloropsis salina is one. So we have to find an alga that is better than our current gold standard Nanochloropsis salina. And the other one was Chlorella protothecoides. Here I compare 2 different Nanochloropsis species. One of them is Nanochloropsis salina, the gold standard strain. And what we find has to be better than that in terms of growth and productivity in the end. And here's one of the best strains that came out of the early screen. You see in blue, start at the same biomass. This one takes off fairly fast. So in terms of Nanochloropsis salina has usually a long lag phase. So this one takes off much faster. Ultimately, the growth rate is the same, except that it's shifted by 3 days. And 3 days mean a lot. And then it goes to a very good density beyond 3 grams. And then we did the lipid extraction. Okay. And we looked at the lipid productivity as well. So we have some strains that in the lab clearly outperformed, at least in the biomass productivity, the Nanochloropsis salina. And some of these we were able to pass on. And here's just the one system shown. Dr. Hüsemann at Pacific Northwest National Lab. He's part of the consortium. He has little raceway ponds in a greenhouse. And the nice thing about this is he can control the temperature, he can control the light, and he can simulate different environments. And that is good because we have production facility in specific environments. But if you want to say, okay, what's the climate here? Can we grow the algae here? We could simulate in his greenhouse the environment that you have here. And then he can tell you, will the alga perform in Illinois or will it perform in Texas? Or do you need Hawaii? So, from here, then they again look at the growth rate, and the growth rate that we should meet are the 20 grams sustained per square meter per day with 50% lipid. Now the question is, what kind of strains are coming out of my screen? Well, we find freshwater is a lot of Synodesmus. The best strains are often Synodesmus. Some kind of Chlorococcalis, not flagellate, but brown and green. A number of unknown green. They're just brown and green, and unless you go into the molecular marker, but even then you often only come back with it's a green. From the marine ones, saltwater is amphora. And then said, okay, that's what we get now. What about when they had the aquatic species program? So I went back into the report. Of course, I read the report before, but now I go back to tell you, what did they come up with? That was in the 80s, 30 years ago? 30 years ago. Here's one example. Arizona State was one site where they did bioprospecting. Oscillatoria. This is the fastest growing ones that they had. Cyanobacteria. Here number third was Chlorococcum, so I'm not that far off. Here's Synechococcus, Cyanobacteria, Oscillatoria. So the top ones were Cyanobacteria. Now Cyanobacteria don't make too much oil unless you genetically engineer strains. That's why I sorted out anything that was Cyanobacterium I didn't count. I didn't even put through the screen. So we don't come up with the cyanobacteria here. But if you look at growth, some of them are really growing well. Then Chlorococcum, and then the next one here is an amphora. So we have similar algae come up as the best performers. Some of them are new that were not in their list. So Cynodesmus, for example, wasn't in there. This was growth. Now when they looked at lipid productivity, the amphora came up on top. So it depends on what you're looking at, growth or lipid productivity. Amphora, several diatoms, but the chlorococcum as well, so they're number 2. But in terms of oil, only 1/3 of the amphora. So we come up with some of the similar strains they had about 20 years ago, and we find some that are new. So it's both. And of course, if my screen wouldn't work, I shouldn't be able to pick up the same thing. So it's a kind of validation for the screen that I pick up similar algae here that were known to produce oil. Okay. Sorry, before we go back, well, what happened to all these algae that they collected, those 300 strains that they had in the early 90s? Most of them are lost. So as you could ask, why can't we go back and get those? Well, because a handful of them are still there in a collection in Hawaii and it wasn't available. Many colleagues tried, So we needed new collections. That's the whole point here. And almost every company goes out and looks somewhere. So summary number 1, the isolation screen is extremely labor intensive, just so that you know. It's not trivial. You need lots of undergraduate students that help with all this work, and graduate students. Then of course screening. You need a rigorous screen for what you want. So it's a multi-level screen. It's not just one size fits all. We have a number, dozens of potential candidates. And the bottleneck right now is putting these outdoors and see if they really perform outdoors. They did very well in the lab. And the other part is that this new system I have, the chromatography with mass spectrometry at the end, is very good for, number 1, see what kind of oils are there, what's the lipid profile. Because the question is also, what kind of lipid is broad? We look at the chemistry and say, what is a lipid? It's not just the phospholipid or the trisucrose, it's a whole range of molecules. Okay. And which one is the best for making oil? The conclusion is that the identification of the new strains can only be the first step. I don't think we will come up with a strain all of a sudden that gives us the 20 grams with 50% oil. This is a baseline. We find some that are really doing well, and then we need to improve on those. And the improvement will contain anything we have. Now, genetic engineering is a difficult term. It comes with politics and comes with everything. But let's go not talk anymore about that part. Let me switch gears a little bit and spend a few minutes on the algae metabolism. So what did I learn out of all this screening and stuff? Well, we have primary metabolites. Primary metabolism, that's photosynthesis, that's everything that lets the algae go through their growth cycle and life cycle. But under stress, we have algae that produce carbohydrates. They switch. There's a metabolic switch, and all of a sudden, a lot of what they make is sugars. Or we have algae that under stress, they switch and they make lots of lipids, triacylglycerols. But there are other algae that make triacylglycerols and carotenoids, and some of these carotenoids are pure hydrocarbons, isoprenoids, a different class of molecule. Or we have some that make major portion hydrocarbons, isoprenoids, and fewer lipids. So ultimately we have a range in that diversity of algae, and this is just for the green here, green algae. There is diversity in the response to stress. Some just die, stress, dead. Or you have some algae with specific properties. And if you're after sugar, ethanol, you want this one here, right? You want sugar. If you're oil, you want maybe this, or that. And we have lots of people that work on this part, so I decided to focus on that one, and that goes, why in the beginning did I choose Dunaliella salina as a model organism and also for metabolism and understand more about metabolism because Dunaliella salina is a halophile, likes high salt, and grows in saturated brine solutions. Doesn't like it, but it grows there. Under best growth conditions, which is about 3 times marine salt concentration, it's nice and green. Under stress, it turns orange and it makes oil, triacylglycerols, and these globules here in the cytosol. But at the same time, you sit in the front, you may see little orange, brownish dots, which are carotene bodies. If you go further, that's one of the main algae. When you go into the pharmacy, you find little bottles with provitamin A coming from this organism. Not for biofuels really, but it's a very good algae to study metabolism and stress metabolism. And that's how I got into this bioprospecting. And several other algae make something similar. Oh, sorry, one back. So when you blow this part up here, the chloroplast, you find the thylakoid membranes and you find these little globules that are full of beta-carotene. That's a relatively recent picture here from Robin Roth that's at WashU in St. Louis. Okay, when you go further, so I want to, for the last decade, I want to try and understand what's going on in that metabolism. And of course, the isoprenoids are good. Beta-carotene is a good fuel. Right? It's a C40 hydrocarbon molecule. It's just worth $1,000 or more per kilogram. You can use it for vitamin A, and it's maybe a few cents if you burn it. So that's the difference here. But it can make fairly large amounts, more than 10% of the cellular dry weight as pure beta-carotene. So if you want to tailor your oil, this may be a good organism to begin with, because it has built-in capabilities to sequester your hydrocarbon. The whole problem about the algae oil business is you need algae that make lots of lipids, but where are those lipids? Usually they are in the triacylglycerol oil bodies. Oil bodies means for metabolism sequestration of molecules. If an organism cannot sequester, you can never find lots of oil, except for if they secrete. So it's either secretion of molecules, and then you have to skim off the molecules, you grow your organism, skim off the oil, or you need organisms that will sequester. And Dunaliella salina, as a metabolic sink, has 2 sinks, one in the cytosol for the oil, and one for isoprenoids in the chloroplast. And that's why I'm very interested in this organism. And it's already a commercial organism, one of the largest algae, microalgae grown in the world, successfully grown. Now, not for oil, for beta-carotene. So we can actually look at those. Here you see very few oil bodies. Here's the stressed ones. Here are some more labeled. We isolate the oil bodies now from the carotene bodies, and we get fairly pure fractions. And the next part is to look at the metabolites in there, look at the proteins in there. I'm part of a consortium that's led out of Los Alamos and PNNL Pacific Northwest National Lab, Dr. Magnuson and Dr. Cushman in Reno, and the Joint Genome Institute is doing the genome sequencing. So we have parts of the genome already, and we have a fairly good transcriptome coverage. So if anybody's interested in that, let me know. It's not necessarily public yet, but we can talk about that. Okay, so here the carotene globules, you can clearly isolate those and the oil bodies that you see. Okay. And there's lots more, but should come to an end now. So basically the summary of the metabolism was focused so far on Dunaliella salina in my lab. We have transcriptome, as I said, limited genome sequence. We have identified a number of genes that are involved in the biosynthesis, in the metabolism of the beta-carotene. We are fairly successful with the cell fractionation, and we're going to send off samples probably next week, the week after, for proteomics, so for systems biology analysis. Okay, so that's where we are with that organism. We have a number of new carcinogenic strains that are different than Dunaliella salina. And some of these may also move on to more metabolic analysis. Because Dunaliella salina especially only accumulates carotenoids in the chloroplast. Whereas if you have other algae, they accumulate, they co-localize. There's one metabolic sink, the oil ones. And the triacylglycerols go in and the carotenoids go in. And that's very different from this one. Okay. So, with that, I come to the end for today, but I hope I gave you a few hints in between. We can discuss more, if possible, the rest of the day. I said lots of undergraduate students that participated, several graduate students, and 2 postdocs. And then, of course, all that wouldn't be possible without collaborators. And I'm very grateful to our consortium, that's the NAABB, and of course, for the Air Force Office of Scientific Research that jump-started all that, even before the Department of Energy got in. So we had a head start to it going. Okay, thank you very much.
All right, so obviously we got a little bit of a late start today. I understand some of you probably have to put down other things, but Dr. Polle will stick around to answer a few questions, and then if you want to talk to him individually, we'll probably take questions for about 5 to 10 minutes, I think, and then we'll go into and talk to him individually after that. But if you need to slip out, just go ahead and move out to the side. We understand. And thanks for coming. And go ahead and open the floor for questions.
Yes.
So I know you said you're trying to skim over the biotechnology applications. Are all of these transformable? Can you actually maybe find ones that are maybe just good at growing and then biotech them to be good at oil?
Okay, your question basically goes into the genetic engineering,
Yeah.
which includes the transformation capability. A few years ago, almost none of the algae could be transformed except for chlamydomonas. There have been very recent reports with all the money going into the algae field finally, there was progress made. We have report for some nanochloropsis species. We have some report, well, Dunaliella has been reported, but skeptical about that. There are some other algae, chlorella, that has been reported that transformation works. So there was progress made. It's still not trivial to have an alga and say, I'm going to transform it. But it's getting better. But you still need effort to put into it. So genetic engineering is still a little tricky.
Probably less expensive, probably less regulatory intensive than say genetic corn. You're just making a biopsy that's not as...
Yeah, so the regulatory side, we don't know yet. Could go either way. And I don't want to make any comments about it. Okay. Then there was another one.
How far is Dunaliella in production?
How far has...
How long it has been in production?
Oh, Dunaliella salina or Dunaliella bardaville? You mean in those ponds outdoors? Dunaliella salina, well, for me it's both the same, but the name is different. Okay. Since the early 90s, so about 20 years.
So what's the common mod that you can get from that one? Is it like close to 15 to 20 grams per liter or something like that?
Oh, Dunaliella salina is grown specifically to make beta-carotene. And if I remember correctly, it's maybe 1 or 2 grams per square meter per day. Because they're grown under stress, so they make all that. So we're far off from what we really need to be viable for oil with Dunaliella salina. And even the experts in Australia, I listened to a talk, said, he's the expert, Dr. Horowitz, one of the 2 or 3 experts in the world on Dunaliella salina. But even he said it's not viable for oil. But we can learn a lot, and I'm coming from the fundamental research side. I'm not just the applied biologist. I'm interested in the really fundamental questions so we can build on something. You can't just come out and say, okay, we build a production facility and have no knowledge or limited knowledge. You need solid foundation, especially if we want to go on and move into metabolic engineering. And I'm not just interested in the oil, there's much more that can be done. in theory. Yes?
Do you think that single strain open ponds are a viable model versus multiple strains, co-cropping?
Yeah, so the question is single strain, the single strain approach at one pond the right model or the best model to go? Maybe not. Like a single crop, you can do it. Yes, we do it all the time. I see when there's summer, okay? But there are some reports that say if you have 2 or maybe 3 or even 4, that they are more robust. And I understand some companies are not taking the single strain approach. So, still remains to be seen. I think the the assemblage of multiple is already working. But, yeah, maybe I have to go back and then say, put the best 2 or 3. The problem with all this outdoor is the season, except if you're in Hawaii or someplace like that. You have seasons. And as you have seasons for your agricultural crops, Germany, we have the summer wheat and the winter wheat. You need, or summer grain, winter grain, we need probably at least a summer strain that is really doing well under summer conditions, and we need another additional winter strain in most regions, because even in southern Texas, I just heard the strain was doing well indoors, and then they put it out, but there was an almost frost day the next day, and that was it. So we need winter, and I think winter and summer strains, and maybe in a mix that you just start with it and then they adjust themselves according to whatever it is outdoors. So that's a valid question, and probably part of the production will be multi-strains and part of the production will be one single strain dominant. Yes?
You showed one slide for strain analysis where some strains they peak pretty early and they started to go down.
Yes.
And others took longer but eventually had higher yield.
Yes.
Do you have any idea, in your opinion, is one strain better than the other in terms of mass production?
Oh, in terms of mass production, when you see this, also whatever I had were batch cultures.
Yes.
And it doesn't mean that outdoors there will be batch culture mode. We just need one mode to test. And the question is, we have the ones that grow slower but ultimately reach the same biomass and lipid, maybe even more. Are they still good? I would say no, because that indicates a long lag period. If you go into a culture outdoors, you cannot afford a long lag period. Some other strain will come in and take over, or something else bad will happen. You need a strain that you inoculate and it will take off. You can't afford the strain inoculation and then not much happens before they ultimately get out. So I ruled out all those strains that have a long lag period. You want something that you inoculate and it comes up fast. I can't afford to throw out my seed and then it takes months and months before the first seedling comes up. It's the same thing. So I would say no. Maybe for a special application where you have to be really careful, but not for, I would say no. Not for the first one to go for.
We'll probably take one more question if there is.
Yes?
Thanks for the talk. So here during the talk, you mainly talking on the list of accumulating algae. But as you mentioned, something like the algae have a different species in terms of the composition. Whether you have like algae with a high, not only the lipid content, but also the carbohydrate content, and also the protein content.
Yes.
And this is up to the alternative algal biofuels, like the alcohol or even the ethanol, etc. So my question is, so what do you think will be the future of the algal biofuels? And then matching that, what do you think is the opening up like innovative algae business screening strategy for that in terms of the composition?
Okay, future of algae biofuels.
I know
Still difficult to say. I mean, we have some companies that claim they solved it all, but I haven't seen them anywhere as money makers. Then we have some companies that didn't really talk too much, and they're now, for example, the fermentation zolazyme seems to be doing well. I mean, Market with their approach was doing well, but if that would have been just for oil, I don't think they would have ever gotten that far. That's with a co-product. So I think it all depends on what will be the harvesting and processing technology. If we go the, what's it called, thermal liquefaction, hydrothermal liquefaction, I don't care what lipid is there, maybe. I just say I need a fast-growing strain with as much biomass so I can't afford the lag period. I just want biomass as fast as I can get it. And then I just convert everything into crude oil. Well, then we have to do the business model. But we need a really fast, good growing strain, regardless of what it makes, biomass. If the engineers tell me all I need is biomass, I don't care what it is as biomass, that strain is going for it. But we have so many possible approaches. I think someone will make it. Some already did, but we may have not heard of it. Or if I knew, I couldn't tell you. That's the other.
Let's go ahead and thank our speaker. Appreciation from our department. Try and get a big coffee cup to make sure that
It'll be good. That's good.
all right, that's good afternoon. Thanks for coming. If you want to talk with the speaker,